Welcome to this presentation of SpatialBench, a new benchmark from Latch Bio that asks a deceptively simple question. Can today's frontier AI agents actually analyze real-world spatial biology data? The paper was authored by Kenny Workman and colleagues at Latch Bio in San Francisco. Let's walk through the key findings. Here's what we'll cover in this poster walkthrough. We'll start with the problem, a growing pile of unanalyzed spatial data, then introduce SpatialBench itself, walk through the main results on model performance, drill into task and platform breakdowns, examine the surprising importance of harness design, analyze agent behavior patterns, and close with limitations and takeaways. Spatial transcriptomics assays like Cinium, Visium, and MERFISH capture molecular state directly within tissue, but the raw outputs are growing in scale and complexity far faster than biologists can analyze them. This creates a discovery bottleneck. AI agents promise to bridge this gap, but existing benchmarks test biological knowledge rather than practical data interaction. SpatialBench is designed to fill exactly this gap. Spatial data analysis requires a unique blend of programming skill, data manipulation with complex and data objects, domain-specific biological reasoning, and tacit scientific judgment, like knowing what QC threshold is appropriate for imaging-based assays versus dissociated single-cell. Current agents are unreliable here. They hallucinate, produce misleading outputs, and fail on spatial-specific tasks. Existing benchmarks like BioAsk test knowledge recall, SWBench test code, but SpatialBench uniquely tests real data interaction with verifiable biology grading. Here's the overview of SpatialBench. The benchmark comprises 146 total tasks, distributed across 7 problem types, with cell typing being the largest category at 39 tasks, followed by differential expression at 26, and clustering at 22, as per Table 9. Tasks span 5 vendor platforms. Seeker leads with 43 evaluations, followed by Visium at 32, Xenium at 30, Atlas Omics at 23, and MERFISH at 19, as per Table 9. Each problem snapshots real experimental data just before an analysis step and pairs it with a deterministic grader. The construction pipeline has 4 stages. First, scientists and bioinformaticians collaborate on end-to-end spatial workflows using novel data from diverse tissue and disease contexts. Second, these workflows are decomposed into key scientific steps: QC, normalization, dimensionality reduction, clustering, cell typing, differential expression, and spatial analysis. Third, each step becomes a problem with a data snapshot and a verifiable, context-specific grader. Fourth, and this is critical, every problem undergoes manual quality control, including trajectory inspection across multiple runs and adversarial testing, with agents instructed to cheat, ensuring answers can't be reached without genuine data analysis. This table shows the full benchmark inventory. The 146 evaluations span 5 spatial platforms and 7 task categories. Notice the heterogeneity. Atlas Omics has no normalization or dimensionality reduction tasks, reflecting the different workflow structures across platforms. Seeker contributes the most evaluations with 43, heavily weighted toward clustering and cell typing. Each platform is paired with specific tissues: Xenium with kidney, Seeker with ovary, Visium with bone, MERFISH with brain, and Atlas Omics with brain and liver. Each SpatialBench problem has 4 components: a workspace with data snapshot data, a natural language task prompt at the scientific level, a structured answer format in JSON, and a deterministic grader. There are 5 grader families. Multiple choice and marker gene precision recall are the most common at 45 evaluations each, followed by numeric tolerance at 36, label set Jacquard at 18, and distribution comparison at 3. 3 design criteria ensure quality: verifiability through automatic checking, scientific durability so answers are robust across methods, and anti-shortcut structure requiring actual data interaction. Looking at the 4-panel aggregate view, the headline finding is sobering. Even the best model, Opus 4.5, achieves only 38.4% accuracy. GPT 5.2 follows at 34%, then Sonnet 4.5 at 28.3%. Gemini 2.5 Pro brings up the rear at 20.1%. But accuracy isn't the full picture. Look at the efficiency metrics. GPT models complete tasks in about 2 steps with 56 to 89 second latency and just 2 to 4 cents per evaluation. Grok variants take nearly 10 steps on average with much higher latency. There's a clear trade-off: Opus 4.5 for peak accuracy, GPT 5.2 for cost-effective performance. Table 1 gives us the precise numbers with confidence intervals. Opus 4.5 achieves 38.36% accuracy with a CI of 31.27 to 45.44, using 2.84 steps per evaluation at a cost of 14.3 cents. GPT 5.2 is remarkably efficient: 34% accuracy at only 3.7 cents per evaluation and 89 seconds latency. The Grok models are striking outliers in step count, nearly 10 steps per evaluation, with max latencies approaching 200 seconds, yet their accuracy remains in the low 20s. Gemini 2.5 Pro is the most expensive at 18.8 cents per evaluation, while delivering the worst accuracy. This is where it gets really interesting. Aggregate accuracy masks dramatic variation across task categories. Look at QC. Every model is below 22%, with several confidence intervals overlapping 0. Compare that to normalization, where models reach 60 to 76%. Model rankings also shift. Opus 4.5 is best at spatial analysis at 52.9%, but Sonnet 4.5 takes the lead on dimensionality reduction at 53.3%. Gemini 2.5 Pro underperforms everywhere, most strikingly on spatial analysis at just 9.8%, more than 20 points below the next lowest model. The key takeaway? No single model dominates across all task types. Performance also depends strongly on the spatial technology platform. Individual models swing by 15–20 percentage points across platforms. Opus 4.5 leads on Atlas X Omics, Merfish, Visium, and Xenium, but GPT 5.2 actually leads on Seeker. Seeker is uniformly the hardest platform. All models score roughly 19 to 31% there, despite having the most evaluations at 43. This suggests Seeker's ovary tissue tasks are intrinsically more difficult. The practical implication is that reliable agents will need platform-aware context rather than one-size-fits-all approaches. This is perhaps the most surprising and practically important finding. The same base model, Opus 4.5, achieves 38.4% with the base mini SpatialBench harness, 48.1% with Claude Code, and 61.7% with the Latch harness. That 23.3 percentage point uplift from base to Latch is larger than the gap between any two base models. Even the Claude Code to Latch jump of 13.6 points exceeds the Opus Sonnet gap under Claude Code. The harness, meaning the tools, prompts, control flow, and execution environment, is not just glue code, it's a first-class determinant of performance. Breaking down harness effects by task category reveals where the improvements come from. The Latch harness provides the largest gains on tasks requiring multiple steps and intermediate programming. Clustering jumps from 33.3% to 65.9%, differential expression from 37.2% to 64.1%, and dimensionality reduction from 51.1% to 75.6%. Even QC, the hardest category, improves from 21.7% to 55.8%. These gains are robust across all 5 platforms, suggesting that effective harnesses provide a general analytical capability rather than task-specific shortcuts. Platform-specific difficulties still persist, however, and will likely require deliberate prompt engineering. Manual trajectory inspection reveals distinct behavioral regimes. Look at the minimal genes column. Opus 4.5 applies a median threshold of 10, which is appropriate for spatial assays where per-cell gene counts are intrinsically low. Every other model defaults to scRNA-seq thresholds of 100 to 200, which is too aggressive for spatial data. This domain calibration difference directly explains Opus 4.5's much higher QC pass rate. For instruction following, Grok models are catastrophic, over 7 format errors per evaluation, with all 119 instances of hitting the 100-step limit coming exclusively from Grok runs. GPT 5.2 produces literally 0 format errors. This figure is key to understanding productive versus unproductive exploration. For Opus 4.5, the blue line, pass rate increases monotonically from 26% at 1 step to 50% at 6 or more steps. More steps genuinely mean more exploration and better outcomes. Contrast that with Grok 4, which shows essentially flat pass rates around 27 to 31% regardless of step count. Those extra steps are just retry thrashing, not productive analysis. GPT 5.2 is interesting. It peaks at 2 to 3 steps with 45.4% pass rate, then declines, suggesting these models are most effective when they solve things quickly and less effective at extended exploration. 2 critical domain calibration insights emerge. First, QC thresholds. Opus 4.5 uniquely applies a median min genes threshold of 10, which is appropriate for spatial assays where per-cell gene counts are intrinsically low. All other models default to scRNA-seq thresholds of 100 to 200, which is far too aggressive and filters out most cells. This directly explains Opus's 25% QC pass rate versus 5% for others. Second, Adata.un's exploration. Opus inspects pre-computed results more often and derives a massive 26 percentage point pass rate uplift from doing so. Grok inspects at similar rates but gains only 4 to 6 points. Finding information isn't enough. Models must recognize its relevance. The authors are transparent about limitations. Deterministic graders, while enabling verifiable evaluation, can lose nuance when scientific judgment is discretized into automatically checkable outputs. The snapshot-based approach captures individual analysis steps, but misses the compounding errors and iterative revision that characterize real end-to-end workflows. For example, revisiting QC thresholds after poor clustering. The vision is for SpatialBench to be the first focused contribution toward a broader benchmark family, spanning major biological data classes, where benchmarks serve as evolving specifications supporting test-driven development. Let me leave you with the 3 key takeaways. First, frontier models remain unreliable on spatial biology. Even the best achieves only 38.4%, with QC and cell typing especially weak. There's substantial room for progress. Second, harness engineering matters as much as model selection. The same model jumped from 38.4% to 61.7% with a better harness, an uplift exceeding the gap between any two base models. Tools, prompts, and control flow should be treated as first-class engineering targets. Third, progress requires joint optimization of model and harness with platform-aware calibration. 15, 20-point swings across platforms mean one-size-fits-all won't work. SpatialBench provides the yardstick and diagnostic lens for this test-driven development. Here are the key references cited throughout this presentation: the SpatialBench paper itself, the foundational spatial transcriptomics works, the agent benchmarking literature it builds on, and the public repository where all benchmark materials, graders, and canonical examples are available. Thank you for walking through this paper with me. The key message is clear. We now have a rigorous way to measure whether AI agents can actually do spatial biology analysis, and the answer right now is not reliably. But the path forward is tractable, involving joint optimization of models and harnesses with platform-aware calibration. I'm happy to take any questions about the benchmark design, the model results, or the implications for building practical biological agents.